we decided to talk about long reads, but since we see more and more the labs, the long reads traveling from the research to the clinical routine, we decided to focus on that journey. We will have 2 talks. Thank you, Dr. Denis Chance, for traveling with us, not only here to Gothenburg, but in the last years. He will show us how he's implementing long read sequencing for clinical diagnosis. So he's already in this journey. And then we will have Dr. Ben Lisfield, our managing director at Limbus, talking about addressing some questions that we can only ask to long reads, because there is more information in long reads than for variant analysis. But before we jump into these talks, I would like to introduce who we are. We are a software development company, a German software development company. We are based at the very north of Germany, right at the Baltic Sea in Rostock. We are actually leading the market in Germany. Here you can see some labs and university clinics that are currently using the software varvis. This software is a cloud-based software designed from the start as a clinical decision support system. So basically, after a sequencing run, you can upload the data and externalize or outsource the bioinformatics. We help you with the filtering and classification up until the reporting. And since last year, the classification and reported are powered by AI. This is because we merged with Nostos. Maybe some of you know this company, and since last year, slowly we have been integrating their AI component into varvis and also the team. So we are very glad. It has been like really smooth, the integration in the technical, but also in the personal level. We are really happy to have the Nostos team among us. Importantly, everything like from the secondary analysis, tertiary analysis, the AI component is certified as a Class C device under the IVDR regulation. We say it's an all-in-one medical device. As I already mentioned, we support, I didn't mention, short reads and long reads from the secondary analysis, tertiary analysis up to the report. We provide a QC monitoring module that I would like to focus in at the beginning. Here is our QC view. So each row here is a sample, and we have different metrics: sex determination, identity check, detection of maternal or bacterial contamination. And every time a sample doesn't meet or doesn't meet the quality criteria, we will flag this so you can go into this sample and check in detail what is happening there. We also provide some other statistics, percentage of duplicates, on-target rate, and provide some graphs where one can see just in a glance, for instance, big chromosomal rearrangement. So it's quite visual and easy to go through. Now I would like to show you a bit the part of the short reads, the SNVs and the CNVs view, and give you a taste of the software. So this is the variant view where you have like the general information of the case. Here, I think you can see, I will point here, we have a trio. Here, the index is affected, the mother is unaffected, and if you would scroll down, you would find the unaffected father. This is a US and a germline case. And here on the right, we have already filtering options. We can create virtual panels that are very easy to create. You can drag and drop CSV files. You can manually create the gene virtual panels or modify them very easily. Here we have the index only. For instance, when you only want to see the variants that were detected in the index, not in the mother, father, or any other relative. And pre-filters are particularly useful for whole genomes. So here you can already see the number of variants that you are filtering. Then we have the variant view. Here, each row, like everything is based on reads. So all the views have like this similar flavor. We have in each row a different variant. And we have a big number of columns. You can decide which columns you want to display and also the order that you want to see them. And you can share this across the different members of the lab. You can filter using the columns in with this the funnel here, but also you can create your own filter buttons that again can be shared by the lab members, so you can streamline your workflow. We provide a prioritization score, an HPO similarity score that is not one-to-one match between the HPOs describing the patient and the HPOs of the variant, but it takes in account if the terms are similar. They doesn't have to be exactly the same, and also it takes in account how specific the terms are. The more specific, the more weight the HPO score will have. And here on the right, we have inheritance filter. For instance, here it's highlighted the compound head, but we also provide, for instance, a carrying screening inheritance filter. When you are running the ion integration, like you want to have the AI supporting your analysis, we provide an automated short listing that is ranked, an gene association column, and we have a dual model of prioritization. We have smoking guns that are, this prioritization is based on specificity, and the clues that is based on sensitivity. When you find the variant that you are interested in, you want to check the details, we provide the variant detail window. Everything in blue is a link out. So most of the information or all the information you will need, it's at hand. We provide IGV and UCSC link outs and other figures and numbers from ClinVar, from Nomad, and even some Metascore. Now, for the classification, we provide a ACMG calculator. When you're running your case with the AI, it will highlight the criteria. So when you see the full button in color is the criteria that was selected by the AI. And when only the letters are in color is selected by AI, but requires human review. The AI is not a black box, so we can actually view and check the evidence that the AI took in account to make that decision on the criteria. And then this is a point-based calculator, so it will one can manually modify the criteria and the strength, and this will automatically calculate the final classification that you can submit for your report, but also to build your own database. Now, we jump to the CNVs. Here is the IGV is integrated. So we see here we are in chromosome 22. We are sitting close to the centromere. And here then we have the duplication and deletion track. So the reads that are the yellow circles, whenever they cross the blue and the red lines, we will be talking about deletions or duplications respectively. So here this red box is actually a deletion. Again, we have a grid down here with the detected CNVs. We provide filter buttons, again, an HPO similarity score, and we have DGB and Decipher integrated. Again, the AI will provide another short listing based on the ion rank and an ACMG-based pre-classification. Since the two next talks will be about long reads, I won't go in the detail. But before we jump into the talks, I would like to take a moment and just remind you who is behind all our work, because sometimes we are not in close contact with the most important people, which are actually the patients. That's why we this year are sponsoring Elena Semichin. She's a mother. She's a double Paralympic champion, and hopefully she will be the next as well. She actually holds the world swimming record, but she's also a rare disease patient. Since she's 12 years old, she lost progressively her sight. And she's also a cancer patient. So I invite you to come to our booth. We have a very inspirational video where she tells her story, and she's really a fantastic woman.